I'm Alex Peterson, and this summer I've been working in Dr. Rita Perlingeiro's lab. My project has been to characterize human-induced pluripotent stem cells from patients with Duchenne muscular dystrophy. Now, these iPS cells are different from embryonic stem cells in that they're derived from adult patients and have been reprogrammed to a pluripotent state. This means that they should be capable of becoming a wide range of different tissues. My job has been to use different techniques to look at and describe 4 iPS cell lines. Then I've compared the findings with the characteristics of an embryonic stem cell control. The purpose of this is to demonstrate that the iPS cells have pluripotency comparable to the embryonic stem cells. This is important so that we can establish the iPS cells are as good as the embryonic stem cells so that they can be used in place of the embryonic cells in our lab's future experiments into therapies that will correct Duchenne muscular dystrophy.
Hi, I'm Monica, and I've been working in Dr. Thomas's lab. I have been working with 2 proteins, phospholambin, or PLB, and cardiac calcium ATPase, or CIRCA. CIRCA is a protein that pumps calcium after your muscle contracts to facilitate relaxation, while PLB, on the other hand, can inhibit CIRCA under certain conditions so that CIRCA cannot pump calcium. However, if CIRCA is not able to pump enough calcium, it can lead to heart failure. So in order to understand this problem more thoroughly, we want to understand the structure of the CIRCA-PLB complex better. In order to do this, I have learned to use site-directed mutagenesis to replace one amino acid on PLB's sequence with a cysteine amino acid, and later add a spin label that will only attach to the cysteine. Then I can use electron paramagnetic resonance, or EPR, to observe the PLB at that specific site. And then hopefully in the future, this can be used to design new ways of encouraging CIRCA to pump enough calcium and therefore assist a failing heart muscle.
My name is Zach Stevens, and I've been working in the Lowe lab this summer. My project has been determining the effect that exercise has on dystrophic cardiac muscle. The muscles of people who have muscular dystrophy eventually deteriorate as the patient grows older, reducing their ability to move. Because of this, treatment of the disease is often focused on maintaining mobility as well as skeletal muscle. However, treatment of the cardiac muscle of these patients is equally important, as nearly 100% of them develop some sort of heart problem by the time they're 20, and many of them eventually die of heart failure. Past research in the Lowe lab has demonstrated that low-intensity exercise reduces some of these dystrophic symptoms in these patients. In order to test whether or not the same effect occurs in cardiac muscle, we had one group of dystrophic mice run voluntarily on wheels for 8 weeks, and one group remained sedentary for that same amount of time. At the end of the 8 weeks, we tested them biochemically and histologically for increased connective tissue and collagen content. And we also tested them for certain hormones, such as atrial natriuretic peptide and brain natriuretic peptide, and certain muscle tissue changes, such as alpha-myosin to beta-myosin within the heart. We are currently still in the process of determining whether or not additional damage occurs or whether benefit occurs.
Hi, my name is Matthew Schreier, and I spent the past summer working in the Dr. Garry lab, studying the transcription factor ETV2 and inspecting the development of mouse cells. Based on the hypothesis that mouse stem cells expressing ETV2 will differentiate either into the endothelial and or the hematopoietic lineage. We did this by a method called lineage tracing, in which mouse stem cells expressing the transcription factor ETV2 were tagged with a green fluorescent protein transgenically, and then by harvesting cells from adult mice, looking at the ones that express this green protein and determining if they were in the endothelial or the hematopoietic lineage. This has a lot of potential in both being able to control the development and differentiation of cells, and it has a lot of potential in the very promising field of regenerative medicine.
Hello, my name is Stefani Prigozhina, and this summer I worked with Dr. Bob Tranquillo in his lab. And for my project, we tested the effects of cyclic stretching on fibrin-based tissue. Basically, we entrapped NHDF cells into fibrin gels in these weld plates. We then stretched the weld plates with a flex cell system for various amounts of time, For example, 15 minutes, 5 hours, 24 hours. After that, we extracted the tissues and we tested the tissues for things like cell and fiber alignment and also the presence of important proteins like ERK and phosphorylated ERK. And the overall goal of my project is to see how we can use cyclic stretching to improve the mechanical properties of tissue-engineered heart valves so we can start implanting those into human patients one day.
Hello, my name is Zhang, and for this summer I've been working in the lab of Dr. Clifford Steer. For my project, I studied the effects of hypoxia on the formation of tunneling nanotubes. Tunneling nanotubes are a recent development in intracellular communication. They are these really thin structures that connect one cell to another and allow for cargo such as proteins and mitochondria to pass between them. For my project, I used colon cancer as a model. I put one set of cells in a low oxygen environment and put another set of cells in a regular oxygen environment, and then I measured the number of nanotubes that formed between the two cells. And my preliminary data shows that hypoxia does indeed increase nanotube formation. This is significant for a variety of disease states such as cancer, for example, in that a low oxygen environment can induce tumors to become metastatic. And my results suggest that tunneling nanotubes could play a role in cell-to-cell communication during metastasis. And also in sickle cell disease, such as the hypoxia associated with sickle cell crisis, tunneling nanotubes could play a role in endothelial cell function.
Hi, my name is Gabriela Ruiz-Colon, and this summer I've been working in the lab of Dr. Joseph Metzger. So the essence of my project stems from the fact that we have a therapy that's developed, but we need an efficient way to deliver that. And so this summer, that's what I've been doing by using a commercial reagent known as Chariot. Chariot has the ability to permeate through cells that are normally impermeable, and we've been able to attach cargo, in this case, the molecule alexafluorphalloidin-488, and we've been able to confirm two things. First, that the cells are still alive because this experiment is being performed ex vivo, and two, that the cargo has actually reached the cell, and we can confirm that by a glow in microscopy. So moving on from this project, we are looking forward to replacing Chariot with the transactivator of transcription known as TAC, which is an HIV gene that is not toxic to humans. And so we'd be able to put therapy that's developed and put it into live human subjects and potentially solve the problem that's not only afflicting our nation, but our world. Thank you.
Hi, my name is Hagen, and I've been working in Dr. Michael Kyba's laboratory. My project this summer has been to differentiate mouse embryonic stem cells into skeletal muscle cells using the gene MESP1. MESP1 is pretty well known in the cardiac world as sort of a master regulator of cardiac development. But recent work in Dr. Kyba's laboratory has shown that it also plays a role in skeletal muscle development. Currently, the Kyba lab uses a MESP1 inducible cell line that is able to be turned on with doxycycline. And they are able to get this cell line to produce skeletal muscle when it would normally only produce cardiac muscle. So my project has been to increase the amount of skeletal muscle that they can get using this cell line because currently the levels are somewhat low at only about 2%. So this summer I've been testing different growth media and passaging techniques and a whole bunch of different small molecule inhibitors in order to try and increase this amount of skeletal muscle produced. This is important because by being able to make more skeletal muscle, we would be able to better understand the process through which MESP1 creates both cardiac and skeletal muscle during development. Additionally, there are a lot of clinical implications with this because we could use this gene in order to produce skeletal muscle to be used in therapies for diseases such as muscular dystrophy.
Hi there, my name is Andy Donaldson, and this summer I worked in the lab of Dr. Garry. The aim of my research was to study the organization of group 3 and group 4 afferent neurons in normal and heart failure animals. The exercise pressor reflex regulates blood pressure and heart rate during exercise. In heart failure, this response is exaggerated, group 3 afferent neurons being hyperresponsive and group 4 afferent neurons having a blunted response. There's preliminary data that suggests that these two groups of neurons are reorganized in heart failure. Therefore, the aim of my study was to investigate the restructuring, or the possible restructuring, of these neurons in the spinal cord, dorsal root ganglia, and skeletal muscle in normal and heart failure animals. Using the scale method, which is a technique used for clearing tissue samples close to transparency, I stained my tissues for TRPV1, which is a protein that is expressed exclusively in group 4 neurons. Preliminary data shows that scale clearing is beneficial for tissue brilliance, clarity, and depth. And this research is important because it could give us a greater understanding of the bodily response and adaptation to heart failure. Thank you.
Hi, my name is Praveena, and I've been working in the laboratory of Dr. James Ervasti. The focus of my summer research project concerns the assembly and disassembly of actin networks, concerted actions that are essential for the generation of force and movement in striated muscle, such as cardiac muscle. I'm currently taking a biochemical and molecular biology approach to study the apparent contribution of cofilin, coronin, and WDR1, 3 actin regulatory factors that are thought together to affect these actin dynamics. I have expressed and purified recombinant forms of these proteins, and am currently investigating their synergistic effect through the use of actin co-sedimentation assays. As a next step, I have also begun to characterize the skeletal muscle phenotype of mice with reduced expression of WDR1. This latter portion of my project involves the use of measurements such as grip strength and whole body tension. Our hope is that insight into the cooperative function of these actin regulatory proteins will provide a better picture of how deficiencies in any one of these cofactors could lead to muscle dysfunction and disease.
Hi, my name is Sophia Zhang, and this summer I've been working in Dr. J. Zhang's lab. My project is called Generation and Differentiation of Human-Induced Pluripotent Stem Cells, or iPS cells. iPS cells are adult somatic cells that have been genetically reprogrammed back to their stem cell-like state, which means that they have the potential to become any cell of a tissue of any organ in the body. So this summer the focus was the differentiation of iPS cells. So we used existing iPS cells from the Kaufman and Slack labs and differentiated them into beating cardiomyocytes. And our future aims are to combine the beating cardiomyocytes with a fibrin tissue, which is biomaterial, to create a contracting tissue equivalent, which we then hope to use to study the mechanisms of heart failure, to test drugs, and to hopefully replace tissue that is lost during a heart attack.
Hi, my name is Jeff Zhang, and this summer I've been working with Dr. Rita Perlingeiro at the University of Minnesota Lillehei Heart Institute. I'm looking at endoglin in the context of cardiac development. Endoglin is a receptor for a large family of proteins that play key roles in both blood and heart development. We already know from previous research that removing this endoglin receptor causes fatal defects in the mouse embryo. I'm looking at the opposite. So what does overexpressing endoglin do? To answer this question, I've overexpressed endoglin in several colonies of mouse stem cells. And then I'm observing changes in these cells using a variety of data analysis techniques. So far, our results suggest that endoglin causes significant abnormal cardiac differentiation and also changes in morphology in these cells. This suggests that endoglin may very well play a very key role in cardiogenesis during embryonic development. Thank you.
My name is Tom Knoedler, and this summer I worked in the lab of Dr. Michael Kyba. So this summer I've been working with growing mouse cells in petri dishes, and these mouse cells that I've been working with are mouse embryonic fibroblasts, so mouse skin cells. And they've been genetically engineered, so we've added 3 genes into them that are engineered in such a way that when we add a chemical called doxycycline, we turn on the expression of these genes. And what we found is that when we turn on the expression of these genes, a small percentage, about 0.1% of these skin cells, are actually reprogrammed into bone marrow cells. What I've been working on this summer is different chemical treatments for these cells to try to increase that conversion percentage with the ultimate goal of getting enough of these bone marrow cells that we can try an engraftment project on a mouse that's deficient in their bone marrow. And this research is important because nationwide there are many people who who are waiting for a bone marrow transplant, but it's pretty hard to match people up for these bone marrow transplants. So these could potentially provide a method of getting lots of bone marrow that's a perfect match for people across the country.
Hi there, my name is Andy Donaldson, and this summer I worked in the lab of Dr. Mary Garry. I forgot my whole script.
This project has been studying the effects using colon cancer cell lines as a model.
Or hopefully bloodborne diseases, as that is
Conditions. Oh shoot, that's okay.
These results are significant because sickle cell disease, such as in sickle cell. Okay.
And then we use the flex up.
We have a great protein therapy, but it's not toxic to live human subjects.
Can be produced using using this cell line. So this is
I'm looking at the context endoglin and key role in the development of the heart. I messed that up. Sorry.
My project is called, oh my god.